Institute of Nanoscience and Nanotechnology in Spain, who will talk to us about electron microscopy techniques and application examples. Before giving the floor to Belén, I would very briefly like to give you some information about Remade@ARI in case this is the first webinar that you are attending. Remade@ARI is an EU-funded materials hub. It aims to assist scientists in both academia and industry and provide them analytical tools to enable them to explore and fabricate and study new recyclable materials. The project has many partners, and through these partners, the project offers access to a high number, 50 analytical research infrastructures across Europe, which give access to a variety of techniques, including synchrotrons, lasers, neutron sources, and so on. The project is currently just over 1 year running, so it's in full swing. We have just closed the second call for proposals for academic research and industry, but pre-proposals can be submitted at any time. So please do visit our website indicated at the bottom of the slide to get more information. And we currently have open 2 calls for SMEs and also for industry in collaboration with knowledge providers. This, as I understand, is a webinar, a part of a series. The idea behind this series is to have speakers from different research structures each month to introduce the technologies that are available in each of the sites. And we aim to teach our members, the partners, the young scientists, but also to inspire users of our platforms. The webinars are usually the last Friday of each month, and you can see in this nice graphic the webinars that have been given so far, and almost all of these are available in the YouTube channel, so you have the opportunity to view them if you wish. As you see, we have a couple of more planned, but we are also intending to follow with the second series in 2024. So now it is my pleasure to welcome Belén to give us a talk about electron microscopy techniques. So I will stop sharing my screen and Belén can start sharing her screen.
Hi. Thank you, Mary, for your introduction to my talk. I will share now my presentation. If I find it here. Okay, share. I hope. Is it visible? Yes? Okay, so then let's start. As Mary was saying, I'm gonna give a talk on the electron microscopy techniques and application examples. Let me see if I can put the pointer. Yes, okay. And I am Belén Ballesteros, and I am the electron microscopy unit leader at ICN2, which is the Catalan Institute of Nanoscience and Nanotechnology. So first, I would like to, let me see if I can, no. Yes, I will leave the pointer. So where are we? We are in Spain, in Catalonia, and close to Barcelona. And in fact, like 30 kilometers inland from Barcelona, as you can see here. And we are located in the campus of the UAB, which is the Autonomous University in Barcelona. So here you can have a picture of the people that are the members of the facility. This is me in the center, and this is the team of research engineers and specialists that work with the facilities that we have. So at the end of the talk, I will give you a brief summary of which equipment we have and which equipment is offered in the Remade@ARI project. So just a bit of historical background. Electron microscopes were developed to overcome the limitations of light microscopes. And this was back at the end of the 19th century, 1900s. So this expression gives this expression that gives you the relation of the resolution with the wavelength of the beam. So back at the time, they realized that if we were using, I cannot use the pointer, and I'll see this way maybe. Yes. Back at the time, they realized that with wavelength, the wavelength of light, which is around 500 nanometers, the best resolution you could get, it's around 300 nanometers. And this is enough to study cells, study bacteria, but it doesn't allow you to discern of smaller things like virus of molecules. So then if you use, instead of light, you use electrons, which has a much smaller wavelength, the resolution, the theoretical resolution, is much far below the angstrom level. So with this in mind, Ernst Ruska and Max Knoll were working and were developing the first TEM in 1931. This is a picture of them working or doing preparing the equipment. And this is an image of some of the notes that they were taking. So this development was such an important breakthrough in science that they won the Nobel Prize for this, together with Ernst Ruska. Well, this was given to Ruska, and they wanted to, together with Binning and Rocher, that developed the STM, which is another type of microscope. So as I was saying, this graph is gonna show you the relation of the time, the years, with the resolution in microscopy. So the blue graphs is giving you the resolution of the light microscope, the conventional one. And as I was telling you, this was limited because of the wavelength of light. And when electron microscopes were introduced, there was a huge improvement of the resolution. So in 1934, it was around 100 nanometers. Just 5 years later, the resolution was improved down to 10 nanometers, and this was for the first commercial TM by Siemens. And then there was a time in the 70s, that they realized that they were working with very high voltage microscopes, and I will tell you in a minute why they were working with high voltage. So they were increasing the voltage a lot in the microscope to reduce, to get better values for the resolutions, and they could go down to 4 angstroms, At some point, this got stable. They were not huge improvements, but again, there has been a new era in electron microscopy, and this is thanks to aberration correctors. And now we are getting to sub-angstrom resolution. So just a graph to exemplify what I was explaining about light microscopy and electron microscopy. So what can we see with our eyes? So we can go down to the thickness of a human hair. That's on the 100 micron range. Below that, you need a light microscope. And you can go down to the 100 nanometers, But if you want to go much lower than that, you really need an electron microscope. And this allows you to study virus particles, DNA, molecules, and even atoms. Okay. So I was telling you, in electron microscopes, now we are not using light anymore, but we are going to use electrons. And there are different types of electron microscopes, as I will explain you, but there are some things that are common to all microscopes. One of them, of course, is the source of the beam. Now we are working with electrons, so all electron microscopes have an electron gun, and this is the part of the microscope where the electrons are produced and they are accelerated to a high potential. So what we need is a large, stable current in a small electron beam. So we have different types of GANs. We have the tungsten filament, which is the most basic one. We can have the lanthanum hexaboride. Both these two types are thermoionic, so you have to heat them to get the electron beam. And also we have field emission GANs, like the one here, okay, which are, if you want to get high brightness and the better resolutions you get with field emission GANs. So the other characteristic which is common to all electron microscopes is that they work under vacuum. Okay, why do we do not? Why we need that? We need the electrons to behave as light. We don't want them to be hitting with particles in their way to our sample. So, and then also the vacuum is needed by some detector and lenses that we are using. And if we work under vacuum conditions, we reduce the contamination buildup. Having said that, there are some microscopes that can work in non-vacuum condition or no high vacuum conditions, like they are called environmental microscopes. But this is like it's not the common. So most of the electron microscopes will work under vacuum conditions. And then the other thing that is common to all electron microscopes is that now the lenses need to be electromagnetic lenses because they need to manipulate the trajectory of the electron beam. So you cannot work with with optical lenses anymore, but with the electromagnetic lenses, you are able to compress, to focus the beam and to direct it to the sample, and also when it leaves the sample to direct it to your detector. So I will talk a bit later about this, but electromagnetic lenses, they have aberrations, and this is going to limit the resolution. And the aberrations, the typical ones are, the most common ones are stigmatism, spherical aberration, and chromatic aberration. So this is going to limit the resolution we are going to get in the microscope. So just one more thing about, I was talking a bit about the spatial resolution, and that we want to improve the spatial resolution, and this means getting a smaller number. No, the smaller it is, the smaller the things you can distinguish when you study when you study them in the electron microscope. So these two expressions give us the relation here. We will get the relation of the wavelength with the resolution, which is kind of the same that I was showing before. So the smaller the wavelength, the smaller the value of the resolution. So, and this means it's better. And on the other hand, this is the relation of the wavelength with the voltage of the electrons, and that the electrons are accelerated to. So the highest the operating voltage of the microscope, the smaller the wavelength. Therefore, the higher the operating voltage, we have a smaller wavelength and we have smaller resolution. So increasing the operating voltage of the microscope will improve our resolution. And I was just saying that in the 70s, they were working with high voltage electron microscopes. They were accelerating the electrons down up to 3 million volts or even 4 million volts because they wanted to improve the resolution, but this had a very big drawback. And the drawback was that at this huge accelerating voltage, you were destroying most of the samples. So we want to find ways to improve the spatial resolution, but without having to increase the accelerating voltage. So about the principles, let me tell you a bit what happens when an incident beam of electrons interact with a sample. So this will be our sample here. And then we will have a beam that interacts with a sample. And there are several physical processes that happen simultaneously when we have this beam interaction. And then we will have different signals that are ejected out of our samples. So we have secondary electrons, we have backscattered electrons as well. We have X-rays, visible light, or rare electrons. And if the sample is thin enough, but only in the cases that the sample is thin, some of the beam will also be transmitted through the sample, and we will have a direct beam. and some inelastically scattered electrons and elastically scattered electrons. So all this is happening simultaneously at the same time. What can we do with an electron microscope? Well, depending on the detectors that we use, we will get the signal, one signal or the other. So if we want to detect the secondary electrons or the backscattered electrons, this is something we normally do in an SEM, scanning electron microscope. And which type of information will we get? We'll get information on the topography of the sample, the morphology, and also, if we use the backscattered electrons, we have information of chemical contrast. On the other hand, if we work in a transmission electron microscope that's going to detect this signal here, we will get information on the internal structure, crystalline structure, also elemental composition and oxidation states if we work with ILS or FDM. And last but not least, we can also use an EDX detector or cathodoluminescence, for instance, and detect these signals here. And we will have information on composition and optical properties. So these signals here, you can get them both in an SEM or in a TEM. So in summary, electron microscopy will give us a lot of information that allows us to understand the structure properties relationship in our samples. So as you probably imagine, there are different types of electron microscopes, as I was explaining. So in the SEM, this one is a scanning electron microscope. Then we have the transmission electron microscope, and then we have dedicated scanning transmission electron microscopes, which are not so common. They are a bit more scarce. No, there are not so many around the world. But this technique, STEM, can be done both in a TEM or in an STM, so with special detectors. So let me tell you about the main differences of the two main types of electron microscopes, which are TEM and SEM. On the one hand, in both cases, we're using an electron beam, but in the case of TEM, this beam is a white beam that's gonna illuminate a huge area in our sample. Well, huge in the micron level, but a whole area in our sample. Whereas in SEM, it's a scanning technique, and therefore we're gonna focus the beam in a very fine point. And this fine point is going to be scanning line by line our sample. I like to explain this in general as if you were entering into a dark room, and you can either switch on the light and then you get an image of the whole room at once, or you can take a torch and you can scan line by line, and when you sum up all the lines at the end, you have the image of the whole room. Okay, so this would be kind of the difference on the illumination in both cases, in TEM and SEM. Then, with regards to the operating voltage, the voltages that the electrons are accelerated to, TEM works normally between 60 and 300 kV. There are some microscopes now that work down to 30 kV, but it's not the most common. Whereas in SEM, the voltages are like between 100 volts or 1 kilovolt kind of, and to 30 kilovolts. So if you remember, I was saying that the higher the operating voltage, the better the resolution. And just from this, you can realize that TEM will always give us better resolution than SEM. With regards of the sample, in TEM, it's quite restrictive because we need that the specimens are very thin. And with very thin, I mean that they have to be below 100 nanometers, depending on the material, but below 100, 150 nanometers maximum. Why? Because we need the electrons to go through the sample. So we really need it to be thin, otherwise they cannot cross. Okay, and not only that, it's not just the thickness of the sample, which is important, but also TEM holders, they have the place where you can put the sample, it's just 3 millimeters in diameter, So this is a picture of a typical grid for TM that you use. And as you say, in this case, it's a copper grid, and it will have a carbon film on top. And then this, you use it, for instance, to deposit nanoparticles. So you can, well, with a solution of nanoparticles, you put, you drop some drops here. And then at the end, you have your nanoparticles on top of this film. But if you see, the size of the sample is like, of the specimen is like 3 millimeters in diameter. If you have samples that are bigger than that, you have to cut them. I mean, otherwise they don't fit. So the sample preparation for TM is much more complicated than for SEM. In the case of SEM, you can have samples that are in the centimeter range, and you just place them on this type of supports, and you put it in the chamber, and the limitation is mainly the size of the chamber. But you can have big samples to put in. Just one more comment. In SEM, if the samples are not conductive, then you need to coat them with some conducting film like platinum, for instance, or carbon. What else? With imaging, I was already explaining it. The electrons need to be transmitted through the specimen, whereas in SEM, the information we're going to collect is near the surface of the specimen. So we are going to get information of the external part of our specimen. And then at the end, in TM, the electrons that are going through the sample, they are magnified to create a real image, whereas in SEM, the beam is scanned along the surface, and the image is built up line by line. So having said that, let me show you some examples of what we can do with each of the techniques, and I think it will be more visible. So SEM, so what we're going to study, the external part of the sample. So we're going to get information on topography, we're going to get information of morphology. This is, for instance, a sample of metal organic framework, so we can study the size, we can study the shape. It's kind of 3D information that we are getting. And also, if we lower a lot the voltage, the electrons do not penetrate so much in the sample, so we get even more surface information. And this is useful to study samples which are very thin, like is the case of this image here, which is of graphene flakes. Okay, so as you know, graphene is just one atom thick carbon layers, and this is really thin, so we could image graphene layers on top of silicon. So yes, the other advantages of low voltage imaging is also that you decrease the charging artifacts. So if you have an isolating sample and you lower a lot the voltage, you don't really need to coat it sometimes. Also, in an SEM, we can get compositional imaging. So if we use secondary electrons, we will have information on the topography of the sample and the morphology. And this is an image of platinum-coated silica. And what we can see is that we have like a spherical particle. And it seems that you have two different things, but well, at least the morphology, the topography is different, no? But if you go to the backscattered electron signal, you can see very clearly that you have two different intensities in the image. So this is much brighter, and this area here, which doesn't seem to be coated, is much lighter. So just with this image, we can know that we have different composition here, and the brighter area will have heavier elements. So we can assign directly that this is where the platinum is, whereas here it's just the silicon. Okay. Some other things we can do dynamic experiments in the SEM, and I will show you some things that we have done in our lab. So we have in one of the SEMs, it's an environmental SEM, so it can work at conditions which are not high vacuum. So we can do we can change the humidity inside our microscope and the temperature. And this is an example of ice formation that we did a few years ago, so that you can see how ice is growing inside the microscope. And in this case, the group, they were interested in knowing the conditions of ice growth on different substrates. So this is the type of study they were doing. And also, we can have like a small furnace in our SEM and do in situ heating and see the evolution of our samples with temperature. So this is a sample of some organic spheres, and this how they look at 25 degrees C. And then if you increase up the temperature up to 150 degrees C, you can see that they are collapsing and they are being destroyed. Okay. So in summary, with the SEM, we're studying the external part of the particles of our samples, and give us information. Also, it can give us information of the composition if we use the backscattered electron detector. Let's go for the TM. So the TM, as I was saying, we are going to have the beam transmitted through the sample. So we have this type of images. We can study, for instance, the morphology and of the sample. So here we see we have these nanorods. So these are gold samples, nanorods, but in some areas we can see that not all the produced nanoparticles are nanorods, but we have some spheres as well. And we can study the internal structure. So for instance, this is a sample of iron oxide coated with silicon oxide. And we can clearly see that inside we have these darker areas which correspond to the iron oxide. And this is something you wouldn't see in an SEM. So what other information we can get? We can get crystallographic information of our samples. So if we have a high resolution TM and we increase the magnification up quite a lot, we can get to see the crystal structure of our materials, and we can start seeing atomic level information. So this is, for instance, a sample of strontium titanate doped with niobium that was grown on top of lanthanum oxide, and we can see the structure of both materials and also the interface, and study if there are defects growing, how is this growth gone, and if the different planes that we're getting, if there are intergrowth between phases, etc. So we can also get this in a TM. And also there is one technique associated to TMs, which is electron diffraction, and this also gives us information on the crystal structure of our sample. And the type of information we get in electron diffraction, we can study or we can know if our sample is single crystalline, and we will have this type of pattern, no, with defined spots. And if we study where these spots are located and the distance between the spots and the center, we can get to know the structure of our material. If we have a polycrystalline sample, instead of these spots, this spot pattern here, we will get this ring pattern, because all the crystals, they are not oriented all in the same direction. And then at the end, instead of having single spots, you have a ring. If the sample is amorphous, you don't get any spots appearing. This is the type of pattern you get. And which other things you can observe. So if the pattern is not properly oriented, you can see that the direct beam is here, and all the spots, the diffracted spots, are here. So you really need to tilt a bit more the sample to have it properly oriented. And if you get a pattern like this, it's because the sample is too thick, and you get to see the Kikuchi pattern. So it's maybe you can study it, but if you really want to go to high resolution, you will need to thin it down a bit more. So let me show you some examples of how a sample appears in an SEM and in a TM. So this is an example of a metal organic framework. how you see it in the SEM. It's a very nice type of particles, which this icosahedral structure. And then in this case, we see the external part, but if we want to see the material that it's encapsulated inside, we really need to go to TM and see what's the internal structure of the specimen. Okay, so these things here would be the platinum nanoparticles. Another example, these are vanadium oxide nanoworkings. This is how they look in the SEM. We see the shape, and then we can see all these filaments that are coming out, that they have a hole. I don't know if you can see it, but they all have a hole. But if you go to TM, you can even see even more. You can distinguish between the different layers and count them and see the size of the hole inside. So you get additional information. Both type of information are really important from the sample. Moving to like biological samples, this is Escherichia coli. So this is a bacteria. This is how it looks in ASTM. It's like more a 3D image, whereas in the TM, this is the type of image you get, which is like a 2D projection of the sample. And one more example on biology. This is pancreatic cancer cell. So this is how it looks in SEM. And then if you go to TM, you can see the internal structure and the ultrastructure of the cell. So I told you about the two main techniques, SEM and TM. Let me tell you about STM, which is scanning, transmission, electron microscopy, and it's the best of both worlds. So we're scanning still, but instead of taking the information that's going up the sample, the secondary electrons and the backscattered, we're gonna take the information that's being transmitted through the sample. And then we have different detectors that we can use, but one of the most interesting ones is the HADAF STEM. So it's high angle annular dark field detector. And why are we interested in this detector? Well, we are going to get Z contrast imaging. So again, we will get compositional images of our samples. So let me give you some examples so that you see why this is interesting. This is a sample of an image of a polymer that has 5 nanometer diameter quantum dots. So they are really tiny. And if we check it in normal TM, I mean, since the image is already in grayscale, it's very difficult to see where the quantum dots are located. If we take this sample and we check it now in HADAF STEM, we have this type of image. So the quantum dots are heavier than the polymers, so they appear much brighter. And now they are very easy to see. And now we can see that they are very evenly dispersed on the polymer. One other example, this is a sample of titanium oxide nanoparticles. It's a matrix of titanium oxide nanoparticles that has some gold nanoparticles. So this is how it looks in the TM. So we have different contrast, the areas that are darker, areas that are brighter. And this is because of the different contrast mechanisms that we have in TM. So we see darker things that are thicker, that are heavier, and also depending on the orientation of the crystals towards the beam, they can also appear darker. So here, as a summary, we don't see where the gold nanoparticles are. If we check this sample in STM with the HADAF detector, now we start to see that there are some areas that are bright, but there are some specific dots that are much brighter. And if we increase the magnification, now we can see clearly where the gold nanoparticles are. So you can see these are the gold, and this is something we wouldn't have seen with normal TM. This is very, very interesting, this technique, when you have different components in your sample that have a very different atomic number. So this is very helpful. Okay, so I told you about these 3 types of imaging techniques that we have, which is SCM, with the different detectors, TM and STM. Then in electron microscopes, we can also do spectroscopy, and we can get to know the elemental composition of our materials. So we have two techniques to do that. One is EDX, and I'm showing an example here. So we were analyzing in these particles which elements we had, and there is iron, titanium, chlorine. So each of these peaks correspond to a particular element. And also, we can do yields. So this is the type of graph that we would get. This background, this is the background here, and this peak here, in this case, is at 708 electron volts, and this corresponds to iron. So these together with the imaging techniques and in STEM, we can do nanoscale chemical mapping. So this is an example of a sample composed by gold and silver. And we can use, in this case, we were using EDX. In STEM, we got where the gold is and where the silver is, okay? So we can do this at the nanoscale. Also, in yields, we can do some other type of experiments. If we study the core loss signal, we can study oxidation states and the coordination. And this allows us, for some elements, to distinguish between different oxidation states and then different phases of the material that we can have. And this is an example with manganese oxide. And also, if we study the low loss area of the spectrum, we can study surface plasmons. So in this case, this was a gold pyramid, and they were checking where the different signals of the plasmons were located. So just to finish, or to kind of, I will tell you a bit of electron tomography. Electron tomography is helpful because, as I was telling you, in TM, we are having a 2D projection of our sample, and this can be very misleading. So sometimes you can think that you have something inside, encapsulated inside, but in fact, at the end, it's not encapsulated inside, but it was on top of your sample, but you're seeing a projection. So what you do in tomography is that you take several images of the sample, you tilt it as much as you can in positive and negative angles. So normally it has to be plus minus 70 degrees, and you take images at, for instance, at every 2 degrees, and you make this type of tilt series. And then there is some special software of reconstruction, and then you can finally, in this case, for instance, we had this particle, this polymeric particle that had some gold metal nanoparticles inside. And after the reconstruction, we could really assure that the particles were inside. Okay, and this is not something you can say directly from a TM image or STM. Okay, so I'm running a bit late in time, I thought, but I want to tell you about aberration-corrected electron microscopy, because this, as I said in the beginning, is the new area on electron microscopy. So, as I said before, the magnetic lenses are imperfect, and these imperfections are the aberrations, and this will limit the resolution in our microscope. So the main type of aberrations are astigmatism, spherical aberration, and chromatic aberration. And astigmatism is something quite easy to correct, so that you can do it in any electron microscope. But recently, well, like 20 years ago, spherical aberration correctors were developed, and this has been a huge improvement in the resolution. So there are also chromatic aberration correctors, but these are not so common. So I will tell you about spherical aberration correctors. So what do they do? So basically, if you don't have, if you have spherical aberration in common electron microscopes, a point in your sample, after going through the lenses, is focused in different places. So those electrons that cross through the middle of the lens will focus in one position, whereas the ones that are going on the external part of the lenses are focused in another position. So you have different focal points, and this gives you a worse resolution a decrease in the resolution on your image. In the case of having zero spherical aberration, a point in your sample is focused in just one point in your image. So that's the best resolution you can get. So a spherical aberration is not something only that only happens in electron microscopes. They had this problem also in astronomy, and they sent, few years ago, they sent the Hubble's telescope to study the galactic nucleus of this galaxy. And when it came back, it appeared they got this type of images, and they were surprised to see, and they were saying, well, I mean, we spent lots of millions of dollars, and this is not the image we were expecting. And then they realized that the lenses had spherical aberration, so they sent a mission to correct for them, and this is the image they got afterwards. So you can see why these are important. So in the case of electron microscope, this would be the image of oxide sample without the spherical, without the corrector. But once it's corrected, you get this type of image, which is much sharper. You get much more detail on the defects, and in summary, you get much more information. So why is this important? Not only you improve the resolution of your images, but also you can get it at lower voltages. So as I said before, increasing the voltage leads to a better resolution, but it leads to more damage in your samples. But what is interesting is to be able to get this spectacular resolution at lower voltages, because then the damage is much lower. So this is the case of lead iodide filled carbon nanotubes. So here the carbon nanotubes would be, it's in dark field mode. The carbon would be outside here, no? And then we see the filling, the material which is inside in bright. But this was done at 200 kb in a conventional microscope. But then this is an aberration corrected microscope at 60 kb, and you can see much more detail of the crystal structure, and in summary, much better information of your sample. So some other examples of things you can do. This is submaximum resolution imaging. This is a graphene sample that has some oxygens, and you can distinguish the oxygens because of the contrast, but also with analysis, and you can study really the defects on the graphene structure. Or you can do atomic scale spectroscopy and atom by atom analyze where you have the different components. So this, for instance, this is showing the strontium, the lanthanum, titanium, and manganese in this sample, in this interface. So, well, I'm coming to the end. I was telling you about all the advantages of electron microscopy, all this information, which is so important to get from the different samples, but it has also some limitations. And I want for you to have them in mind. One of them is sampling. So when you are studying a sample in the electron microscope, we are studying just a very small portion of the sample. So before jumping into conclusions, you should do some statistics. You should check different areas of the samples, and also you should correlate the results you are getting with other techniques which are bulk, and see if it fits. And yeah, be careful, because it's easy to get an image and say, I got what I wanted, but maybe it's not representative of the whole sample. Also about interpretation, different physical processes are happening simultaneously, and they are contributing to the contrast of your image. So you have to bear in mind what type of sample you have and what type of contrast you can get before yeah, again, reaching to some conclusions. And also in TM, as I said before, we have a 2D projection of a 3D sample, and this can be misleading. So you have to have this in mind. With regards to specimen preparation, the sample must fit in the microscope holders, and this is especially especially important for TM. And also, we should try to make it as minimal as possible to avoid any artifacts on the sample. And also for SEM, we have to take into account that depending on the SEM we're using, we will need to coat our samples if they are isolating. And last but not least, we are sending, in the electron microscope, we are sending a beam of electrons to our samples. So the electrons can interact with the sample, and they can change the structure, and they can change the chemistry, because they are electrons, and they can produce reactions. So we have to have this in mind. And also that there are different types of damage that you can get. There is radiolysis, there is knock-on, and there is heating. Each of these types of damage are more prone to happen in different types of samples. So you have to know which are the best conditions to study your sample, and try to work in low dose techniques or change the voltage to minimize the damage. So in summary, this is a sample of field nanotubes, and just some information we could get. We could get information of the morphology. So it's a nanotube, so it looks like spaghettis, if you want it. If we go to HALAB STEM, we can see the internal part, which is brighter, it's filled. If we do it in an aberration-corrected microscope, we can get the crystalline structure of our filling material and much more detail. We can also study in high-resolution TM, and we will have this type of information. We can do EDX and see the different components we have in the sample. And for instance, we could go to EELS and study the electronic structure of our sample. So just a quick mention to the last advances in electron microscopy. So I was mainly focused on spatial resolution and contrast, that it has been improved a lot with aberration correctors, but there are some other techniques that have been developed to improve this, but also improvements in energy resolution thanks to monochromators. You can also study physical properties in the electron microscope. There are holders that allow you to study the optical, the magnetic, and the electric properties in your materials. Not only you can do it in static, but also in situ. So you can change also the environment, the temperature, and even work in liquids, and study your sample in conditions which are more close to real conditions. And this, for instance, is very interesting as among others in catalysis, for instance. And then there is the development of low dose techniques like cryo-TM, IDP system, etc. And also there are many much research going to get 3D information and to get atomic scale quantitative information on the three-dimensional, on the 3 dimensions, let's say. So just to finish, just to mention which type of equipment we offer in Remate from ICN2. We have an environmental SEM that we use for study of morphology, topography, and also composition of the samples, because it has an EDX. But also we can do in situ heating and in situ humidity experiments. We have another SEM which is even higher resolution, and this one allows us to work down to 100 volts. And normally we work at 1 kilovolt, and we have a lot very nice surface information of our samples. Then we have a conventional TM, Technai F20, that is a high-resolution TM. You can do Z-contrast TEM, chemical analysis with EDX, EELS, and structural determination in 3D. And then recently, 1 year ago, we installed a new aberration-corrected microscope in the facilities of Synchrotron Alba. This equipment was acquired within a consortium that with all these members, and these are the main characteristics, but we can get sub-angstrom resolution even at 60 kb. Has a monochromator. We can do atomic scale spectroscopy. We have a direct electron detector, a very nice EDX and EELS, and possibility to do 4D STEM, etc. And last but not least, as I said before, sample preparation is very important. So we have a sample preparation lab with all this equipment that allows you to prepare samples to use both in the SEM or in the TM. And recently, and with the partnership, the consortia that we bought the Spectra microscope, we also acquired a FIB to prepare lamellas for TM. Okay, so these were some examples of things we can do in the Spectra, but I think I took a lot of time already, so I will leave it here, and I'm now open for your questions.
Thank you very much, Belén, for a very interesting talk. Please, if there are any questions from the audience, you can type them in the Q&A or in the chat, and then I will read them out to Belén. So in the meantime, I have a question, Belén, if I may.
Yeah, sure.
About EDX, is this done in both the SCM and TEM? So
Yes.
do you get information on the surface or only also in the structure of the sample? Okay, so in EDX, what you have is that you're detecting the X-rays that are scattered out of your sample after so the electrons interact with your sample, and then there are these transitions of electrons within the atoms, and then when they relax, they will eject an X-ray, and this is what you are detecting. So this has information on the atom and the nature of the atom that you are detecting. And as you said, you can have it both in the SEM and the TM. The information you get is not just of the surface, so it penetrates like several microns inside the sample. The thing is that in the TM, because the sample is so thin, so you will have information of all the thickness of the sample, but the sample is very thin, so it's mainly, I mean, it will not change so much. I mean, you have to have in mind that it's not just the first nanometers, but it's the whole 100 nanometers thickness. But in the SEM, this is very important, because if you are interested in analyzing, for instance, the top layer in a sample, and this layer is like, I don't know, 500 nanometers, and you send the electrons at, I don't know, 30 kb, you will also get information of the substrate. Okay, you can change the amount, so the thickness of let me see how I say it. So you can try to improve or enhance the amount of signal from only the surface by changing the voltage. So if you go to lower voltages, your electrons are not penetrating so much, so your EDX signal will come more from the surface. I don't know if it was clear, my explanation. Yes, very clear, very clear. Okay. Thank you.
Okay.
I don't see any other questions at the moment. No. In the meantime, I will ask the attendees. If possible, I will type a little survey link that we have. And if you have a couple of minutes, you can answer some questions, very 2 or 3 questions. And this is particularly interesting at this time because, as I said, we are planning the next series. So there is a question there on what you would like to see in this webinar series. So please, any suggestions are most welcome. Okay, so I don't think there is any more questions for Belén.
Okay.
Again, Belén, thank you very much for this
Okay,
very interesting
thank you.
talk. And if you can please stop sharing your slides.
Yeah, sorry. Yes, sorry.
No problem. I just have one final slide to show, just to let people know of our next webinar. So our next webinar is scheduled for the 24th of November. We will have Iva Bogdanovic-Radovic from Rude Boskovic Institute in Croatia, and she will talk about the ions as a powerful tool for analyzing and modifying materials. This is on the 24th of November. And then this will be the last of this year, and we will start again in January. So thank you very much, everybody, for being here today. And I'm wishing everybody a nice weekend.
Okay, thank you. Have a nice weekend.
You too.
Bye. Bye.